students, welcome back. In the previous videos, we covered the topic of monohybrid cross inheritance. Now we will explore the concept of inheritance of two genes. Sir Gregor Mendel crossed plants with two different characters. For example, he crossed two plants, one with round seeds and yellow color and the other with wrinkled seeds and green color. Capital R and capital Y are the genotype of round and yellow seeded plant. Small r and small y are the genotype for wrinkled and green seeded plant. In F1 generation, we will get round and yellow seeded plants only, that is with capital R, capital Y and small y. Let's study the F2 generation using a Punnett square. Here capital R and capital Y are the dominant alleles for the round and yellow seeded plants, and small r and small y are the recessive alleles for the wrinkled and green seeded plants. We have 9 round and yellow seeded plants, 3 round and green seeded plants, 3 wrinkled and yellow seeded plants, and 1 wrinkled and green seeded plant. The phenotypic ratio is 9:3:3:1. In monohybrid crosses, we studied that gene inheritance follows the principle of law of segregation. The inheritance of a dihybrid cross, on the other hand, relies on the principle of independent assortment. According to the law of segregation, the alleles separate and segregate from each other independently during the gamete formation. If there are parents with yellow and green colored alleles, then there is a 50% chance for one gamete to get capital Y and other gamete to get small y. Law of independent assortment states that when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair of characters. To understand the law of independent assortment, let's check the previous example one more time. Our F1 generation was capital R, capital Y and small y. During gamete formation by meiosis, there are equal chance that the gamete gets capital R, capital Y, capital R, small r, capital Y and small y. The inheritance of capital R is nowhere linked to the inheritance of capital Y. They are inherited separately. The yellow color is not linked to the round shape of the seed. Yellow color can be combined with wrinkled seeds as well. Both the genes coding for these characters are inherited separately and independently. That is why we get the new traits in the F2 generation that are different from the parents. Let's learn chromosomal theory of inheritance. Mendel's groundbreaking research on character inheritance was published in 1865. But it took until 1900s for his work to gain recognition from the scientific community. This was because of some reasons. During that period, communication was quite challenging. According to him, the pair of alleles did not blend with each other. This cannot explain the variation in offspring from parents. If alleles did not blend, then there would be no variation, but that is not true. He used mathematical expressions to explain biology, like the genotypic and phenotypic ratios, which was not acceptable to his contemporaries. The final reason was that he failed to provide any evidence to support the existence of factors, that is, genes. After 1900s, there was advancement in microscopy and scientists could study more about cell division. During 1902, two scientists named Walter Sutton and Theodore Boveri were studying about chromosomes independently. Sutton was experimenting on grasshoppers. His works showed that the chromosomes occur in matched pairs of maternal and paternal chromosomes. That means there are homologous chromosomes in which one we get from mother and one from father. He said that these matched pairs of chromosomes separate during meiosis. Boveri was studying sea urchins in which he found that all the chromosomes had to be present for proper embryonic development. They used their knowledge of chromosome movement to explain the Mendelian principles. We know that meiosis or reduction division happens in the formation of gametes, that is in the egg and sperm cells, the number of chromosomes in gametes has been reduced to 23 from 46. That means gametes are haploids. Two haploid cells join to form one zygote, which means 23 chromosomes from each haploid cell form the diploid cell, forming the zygote with 46 chromosomes. The chromosomes occur in pairs and are called homologous chromosomes. Among these, we get one from mother and one from father. Similarly, same gene or alleles of the same gene are present in the homologous chromosomes in the same site. These sites are called locus. The genes or the alleles of the genes are present in the same sites in homologous chromosomes. As we know, meiosis has two phases, meiosis 1 and meiosis 2. In anaphase of meiosis 1, the paired homologous chromosomes are separated to the poles by the spindle fibers, but still the sister chromatids are joined together. During anaphase 2, the sister chromatids get separated from each other by the spindle fibers to the poles. Now they are called chromosomes. At the end of telophase 2, there are 4 haploid daughter cells are formed. Sutton and Boveri argued that the pairing and separation of a pair of chromosomes would lead to the segregation and separation of the factors. They explained the Mendelian principles based on their findings and called it the Chromosomal Theory of Inheritance. What we see is the usual process of meiosis. However, during prophase 1, the homologous chromosomes from each parent pairs up to form a tetrad. That is, 4 sister chromatids will be there. During this time, there are chances for crossing over. Crossing over simply means the switching of chromosomal parts. That means parts of the chromosomes that contain genes can be switched. So the chromosomes in the 4 daughter cells will be different from that of the parental type. And this is the explanation of variation. Here you can see these haploid cells. They are different from their parent cell because of crossing over. Later research conducted by Thomas Hunt Morgan on fruit flies, particularly Drosophila Melanogaster, revealed the mechanisms through which genetic variation arises as a result of sexual reproduction. In order to research sex-linked genes, Morgan conducted multiple dihybrid crosses on Drosophila. Sex-linked genes refer to characteristics or traits that are influenced by the genes present on the sex chromosomes. Morgan hybridized yellow-bodied white-eyed females to brown-bodied red-eyed males. The alleles of these genes were present in the X chromosome. The gene for body color is denoted by Y and gene for eye color is denoted by W. In females, there are 2 X chromosomes, that is why YY and WW. Y and W are recessive genes. In male, there are 1 X chromosome and 1 Y chromosome. The gene in X chromosome of male is Y plus and W plus, which are dominant. Here the recessive allele is represented by the alphabet and dominant allele with a plus sign on the alphabet. The alleles in female parent were recessive and in male parent were dominant. In F1 generation, Morgan got females and males. The X chromosome from female joined with the X chromosome from male to form XX, that is female. Here, the female gets the dominant alleles and becomes the wild type, that is brown-bodied and red-eyed ones. The X chromosome from female and Y chromosome from male join to form XY, that is male. The male offspring get the recessive genes and became yellow-bodied and white-eyed one. The F1 generation genes were the parental type. Now let's understand the gametes formed by each of them in F1 generation. In female, one gamete contains the recessive Y and W. Second one contains the dominant Y plus and W plus. In third gamete, crossing over between Y plus and W happen and the resultant genetic combination will be Y plus dominant and W recessive. In the fourth gamete also, crossing over happened between Y and W plus and the resultant will be recessive Y and dominant W plus. Here two gametes are parental type and two are new, that is they are recombinant ones. In male there are two gametes, one that resemble the parent X chromosome, that is recessive Y and W, and the Y chromosome. Now let's understand F2 generation. Here we can see some recombination, the combination of genes which are not present in the parents. Genetic recombination is when genetic material is exchanged between different organisms, resulting in offspring with different traits than their parents. In F2 generation, X chromosome from mother joins with X chromosome of father where Y and W are recessive. This fly will resemble the father, that is yellow body and white eye. Next, this X chromosome that has dominant alleles from mother joins with X chromosome of father that has recessive alleles. The resultant offspring will be wild type, that is brown body and red eye. These two offsprings are parental type. Now the recombinant ones are formed by joining the rest X chromosome from each parents. Here we get brown-bodied white-eyed fly and yellow-bodied red-eyed fly which are different from their parents. They are the recombinant ones. The male offspring are formed by joining X chromosomes from mother and Y chromosome from father. The first one become the parental type with yellow body and white eye. Second one also resembles the parent with brown body and red eyes. Third one is the recombinant one with brown body and white eye, and the last one also recombinant with yellow body and red eye. But here the parental types were more with 98.7% and recombination types were less with only 1.3%. Here if you observe, you can see that the genes are close together. Here you can see in a chromosome, the genes are close together. That means they are linked to each other, and that is why the chances of recombination were less. They are inherited as one. This is called linkage of genes. But if the genes in a chromosome are far apart, that means they are not linked together. And then chances of recombination is very high. If genes are far apart, recombination will be high. To prove this, Morgan crossed one white-eyed miniature winged female with a red-eyed normal winged male. The alleles in female were recessive with W for eye color and M for miniature wing. The alleles in male were dominant with W plus for eye color and M plus for normal wing. The F1 generation he got were all wild-type female with red and normal wings and white-eyed and miniature winged males. In F2 generation, as in our previous example, Morgan got parental types and recombinant types. However, as the alleles were far apart, the recombinant types were more than the previous one. Here you can see these genes are located far apart in the chromosome. As a result, here the parental types were 62.8% and the recombinant types were 37.2%. Here the recombinant types are more than the previous example. Morgan's experiment showed that when two genes in a dihybrid cross are on the same chromosome and they are close to each other, the proportion of parental gene combinations are greater than the non-parental type. If you like this video, please share it with your friends. If you have any queries, put it in the comment box. Thank you.